Another study looking at the cellular phenotypes is from Anne Bang, who is at Sanford Burnham Institute, and she will be developing a suite of assays for the analysis of PWS patient iPS-derived cortical neurons. So rather than using the CRISPR neurons that the other group is using, she's going to use patient-derived neurons that were developed by Stormy Chamberlain's lab at University of Connecticut, and she is going to apply assays. Dr. Bang is very experienced in high throughput assays for neurons, so she's going to take that expertise and apply it to PWS. So they'll use, and this is the first step towards screening for drugs, right? Screening for new drugs that might treat PWS. So important throughout the brain. So we're excited about this study because it brings an experienced cell biologist who's at a premier drug screening institution who kind of does this, all the time to look at our PWS cells and really, apply their knowledge to that. The studies are going to provide a basis for understanding cognitive and psychiatric challenges in PWS. That's what cortical neurons are really important in, that thinking and mental function. And the goal is to develop rigorous cellular assays, which will then lay the groundwork for doing drug screens to see if there are ways, if we do find changes in the cortical neurons, are there drugs that can kind of encourage them to be more typical in their functioning. So the goal is to develop a validated PWS assays to be used in drug discovery efforts and eventually to actually do that screening.